although DNA contains the genetic information that is used by the cell to synthesize the many different proteins that are used by that cell, the DNA molecule itself is not directly involved in protein synthesis. In fact, what the cell does is it uses an intermediate molecule known as RNA, which is involved directly in protein synthesis. And as we discussed in the previous lecture, there are 3 types of RNA molecules, and each type of RNA molecule serves its own unique purpose in protein synthesis. Now, we see that the DNA is basically first transcribed into our RNA. So we undergo the process of transcription in which we synthesize RNA from DNA, and then the RNA molecule is directly involved in the process of protein synthesis. So in a way, we can imagine that the process of transcription is the passing down of the genetic information from the DNA molecule to the RNA molecule. Now, the question is, why can't we use our DNA molecule to directly synthesize our proteins? Why do we have to use these intermediate RNA molecules? Well, the answer is pretty simple. So the advantage of using RNA in that cell is to basically make sure that we do not damage or hurt our DNA in any way. So we want to ensure that the DNA is not mutated, because the DNA is essential for the survival of the cell as well as of the offspring. So during reproduction, it's the DNA that is passed down and not the RNA. And so that means we have to make sure that the DNA is not damaged. And by using these RNA molecules, we ensure that the DNA basically decreases the opportunity to damage itself. So we see that because our RNA is not passed down to the offspring, our cells have no problem recycling and breaking down the damaged or mutated RNA molecules. So we see that transcription is the process by which we pass down the genetic information from our DNA molecule to our RNA molecule. And because DNA are only found in 2 locations, in the nucleus and the mitochondria, we see that the process of transcribing our DNA to RNA only takes place either in the nucleus or in our mitochondria. Now, the process of transcription can be broken down into 3 stages. We have initiation, we have elongation, and we also have termination. So let's begin by discussing what initiation is. Now, just like in the process of DNA replication, the first step in our transcription process is to actually find that initiation point that basically is the location where we unwind our double-stranded DNA molecule. Now, a group of proteins known as initiation factors basically move along our double-stranded DNA molecule until it reaches a special location, a special region on the DNA known as the promoter region. Now, what the promoter region is, it's basically a special sequence of DNA nucleotides that signals the RNA polymerase to actually join the protein complex and begin the synthesis of our RNA strand. Now, the promoter region usually does not vary too much from one organism to another organism. In fact, the most common type of sequence that appears in our promoter region is known as the consensus sequence. And we'll talk much more about the consensus sequence and this promoter region when we'll get into the field of biochemistry. So let's discuss what we just said by looking at the following diagram. So basically, this is our double-stranded DNA. So this is the 3' end, this is the 5' end of the single strand shown above, and this is the 5 and this is the 3' end of the single stranded DNA molecule shown below. And they're connected via hydrogen bonds between our nitrogenous bases. Now, this is our RNA polymerase as well as our initiation factors. And as the initiation factors move across, eventually they come across a region, a sequence of DNA nucleotides known as the promoter region that basically signals our RNA polymerase to begin unwinding and to begin the synthesis process. So when this reaches this region, basically the RNA polymerase combines with this initiation factor, with these proteins, and it's the RNA polymerase itself that actually unwinds these two molecules, unwinds the two single-stranded DNA molecules, and creates a bubble that is known as the transcription bubble, and we'll discuss that in detail in just a moment. So basically, one important difference between DNA polymerase and RNA polymerase is that DNA polymerase itself cannot actually unwind the double helix of our DNA, but the RNA molecule, the RNA polymerase, can in fact itself unwind our molecule. Recall that in our discussion on DNA replication, a special type of enzyme known as DNA helicase actually has to unwind the double helix, but in this case, the RNA polymerase itself can actually unwind our double helix. So this concludes our discussion of initiation. Let's move on to the process of elongation. So elongation is basically the process where our RNA polymerase actually synthesizes the RNA molecule. So once RNA polymerase unwinds the DNA, it creates a bubble that is commonly known as our transcription bubble that is shown in the following diagram. So basically what the transcription bubble is it's a region where our two single strands of DNA have been separated and are now exposed. Now notice only one of these DNA strands is actually used to synthesize our RNA molecule. So to see what we mean, let's take a look at the following diagram. So we can imagine that our initiation factors, these proteins, are moving across our DNA. Eventually, they reach our promoter region, and then the RNA polymerase joins and it unwinds our double-stranded molecule, our double helix, and it creates this transcription bubble. And now, as our RNA polymerase moves across this upper strand, which runs from the 3 to the 5 direction, it uses this strand to basically synthesize our RNA molecule. So it uses this strand because just like DNA polymerase, RNA polymerase can only read our DNA strand beginning at the 3' end and ending at the 5' end. So it synthesizes our RNA beginning at the 5' end and ending at the 3' end, as shown in the following diagram. So this green strand is in fact our RNA strand. Now, the DNA strand that is used to actually synthesize the RNA strand is known as the template strand, or more commonly, the antisense strand. Now, the other DNA strand is called the sense strand or the coding strand. And basically, the nucleotides found on this strand are almost exactly the same as the nucleotides found on the RNA strand. The only difference is the thymines are replaced with uracil on the RNA, because remember, in RNA, the thymines are replaced by the uracil. So basically, the reason this is called an antisense, because this has a complementary nucleotide sequence to this strand here. So this 3 to 5 strand is known as the antisense strand. And now RNA polymerase uses the antisense strand to basically direct the synthesis of our RNA molecule. And the other strand is known as the sense strand or the coding strand. So Once again, notice that just like DNA polymerase, RNA polymerase can only read the DNA strand in the 3 to 5 direction, and it can synthesize the RNA strand shown in green in the 5 to 3 direction. Now, another important distinction that must be known between DNA polymerase and RNA polymerase is the following. DNA polymerase has the ability to basically correct any mismatches that are made with our base pairs. However, RNA does not have this proofreading mechanism, and that means many more errors are actually formed during the process of transcription than the process of replication. And that makes sense because during replication, we have to replicate our DNA precisely to pass it down to our offspring. Now, the other thing that I want to mention about RNA polymerase is the following. In prokaryotic organisms, there's only one RNA polymerase, while in eukaryotic organisms, there are 3 different types of RNA polymerases, and each one of these RNA polymerases is used to synthesize the 3 different types of RNA molecules: our messenger RNA, the transfer RNA, and the ribosomal RNA. Now, what about the process of termination? So, just like there is a sequence of nucleotides that initiates the process of transcription, there is also a sequence of nucleotides, of DNA nucleotides found on the double-stranded DNA that also signal the process of termination. So the process by which our transcription basically stops. And the sequence of DNA nucleotides is known as the termination sequence. Now, when the RNA polymerase basically reaches this termination sequence, certain types of specialized proteins may assist in the process of termination. For example, in prokaryotic organisms, a special type of protein known as the Rho factor binds to our RNA and helps the RNA to dissociate from our DNA molecule, and that basically terminates the process of transcription. So once again, transcription is the process by which we basically transfer the genetic information from DNA to RNA. And there are 3 stages of transcription. We have initiation, we have elongation, and we have termination.